Hello guys, now we are going to start microbiology. So let's start. So who is the father of microbiology? First thing first, who is the father of microbiology? It's Louis Pasteur. Okay, and what does Louis Pasteur give? Louis Pasteur give us that germ theory, and he introduced some principles, like he introduced principles of sterilization and disinfection. Okay, so the second name in the list is Joseph Lister. Remember, he introduced principles of asepsis. Asepsis means on some surfaces or at some place there is no microorganism present. Okay, so asepsis in surgery. Okay, now the third name in this list is Robert Koch. Okay, so Robert Koch basically tells us about some organisms at that time. The first one is TB and the second one is cholera. Okay, tubercle bacillus. Okay, now the second point which we will discuss now is about cell wall. Cell wall. You know, cell wall is made up of amino acids like diaminopymalic acid. Okay, diaminopymalic acid. Now, in this cell wall, let's discuss about prokaryotes. Okay, so in prokaryotes, the cell wall is made up of basically N-acetyl murmuric acid. What? N-acetyl murmuric acid. And the second thing is N-acetyl glucosamine. Okay, you know what are prokaryotes? Prokaryotes have basically no cytoplasmic organelles. You can write they have no cytoplasmic organelles. Okay, like they basically also don't have no nuclear membrane. Okay, now in this list prokaryotes are done. Now after that, let's discuss about gram positive and gram negative. So first one is gram negative bacteria. Okay, we will discuss about their cell wall. So in gram negative bacteria, cell wall contains lipopolysaccharide. Lipopolysaccharide means endotoxin. We also call them endotoxin. Okay, and periplasmic space, and as you all know, outer membrane. Okay, that's it for gram negative. Now let's go to gram positive. Gram positive bacteria, the important in them in gram positive is, just remember, the gram positive bacteria cell wall have thicoic acids. What? Thicoic acids, and they don't have outer membrane, as we all know. Okay, so after this, let me tell you something about endotoxin, endotoxin. Okay, and the second side is exotoxin, exotoxin. Okay, so this endotoxin is you can found only in gram negative. Okay, and they are basically made up of lipid complex. Okay, that lipid A. Okay, lipid complex. They are heat labile. Okay, basically they are heat stable. Okay, they have low antigenicity. Okay, and they usually are secreted, or they are usually formed in our body on the lysis of bacteria. Okay, so they are formed on the lysis of bacteria. Okay, whenever bacteria lysis is there, bacteria break, then this endotoxin is released. Now exotoxin, this exotoxin is present in both basically. Okay, it present in both. Both means gram positive and gram negative, you can write. Okay, they are very highly antigenic. They are very highly antigenic and they are actively secreted. Okay, they are actively secreted. Now write down one note, it's important note. Okay, the botulinum, this botulinum, this botulinum toxin, okay, this is only exotoxin that is released on lysis of bacteria. Okay, so whenever botulism will they will break. Okay, so at that time this botulin toxin is secreted. Okay, or release. Now mycoplasma, remember this about this mycoplasma. Okay, this mycoplasma, this is basically, it do not have cell wall. Okay, so it lacks cell wall, and it have sterols in cytoplasmic membrane. Okay, now one more point, spore forming bacteria. Spore forming bacteria. So the spore forming bacteria are like Bacillus and Clostridium anthrax. Okay, Clostridium and Bacillus. This spore, they are resistant to heat. They are resistant to heat means they will not break by heat very easily, because of that there is one acid in them, dipicolinic acid. What does this do? It will make them resistance to heat. Okay, so this all in this introduction.